Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this course, Cell and Molecular Biology, we are discussing about the different aspects of the how the molecules are interacting with each other and what are the different types of interactions are responsible for the different types of physiological processes. So, in this context, so far we have discussed about the evolution, we have discussed about the origin of life, we have discussed about the central dogma of molecular biology, we have discussed about the signal transductions, we have discussed about the uptake of molecules and so on. And in the previous module, we have discussed about the immunology, and in the current module, we are discussing about the different types of topics. So, if you recall, in the previous lecture, we discussed about the gene therapy, and now in today's lecture, we are going to discuss about the cell as a production factory. Now, the mammalian cells or the bacterial cells are capable of synthesizing the different types of proteins, right, and they can be exploited for different production of the different types of proteins. Now, you can see that there is a clear cut comparison, or there could be a similarity between the cell and as well as the factory where you are actually going to produce the different types of products. So, you have the cell membrane, right. So, cell membrane is just like a wall of the factory, right. So, if you see a cell, right, you have a cell where you have the cell membrane, you have the cytoplasm. So, cytoplasm will behave like a floor on which you are actually going to build the different types of machines, you are going to put the machine. So, it is actually going to behave like a floor. And then we also have the some of the the empty areas, and these are being called as vacuole in the case of cell, whereas the same is being used for storing the the different types of products. So, that is being a part of reservoir. Then we also have the different types of machines, right. So, one of the machine is being called as ribosome, which is actually the main machine for protein production. And then we also have the once the product is being formed, then it has to be packed. So, then for that within the cell we have the Golgi bodies, whereas in the factory we have the packaging plant. Now, compared to here, once you are doing the factory, you also require the controlling machinery. So, you actually require the operation center. So, that is being that the same job is being done by the nucleus within the cell. Then you also require supervisor, you also require a person who actually going to give the different types of instructions. So, this job is being done by the DNA within the cell, whereas it is being done by a supervisor in the factory. Then you also require the the energy source, so that you can be able to use that for running the machines. So, that is the job which is being done by the mitochondria, whereas this is being done by the powerhouse. Then once you are produce the product, you are also going to have the byproducts, and these byproducts are going to be taken care by the lysosome within the cell, whereas the same is being done by the disposable system within the factory. And at the end, you are also going to have the the distribution of these packed material. So, you are going to have the manufacturing plant, and you are going to have a distribution center. So, that is being done by the endoplasmic reticulum within the cell. So, what you see here is that the cell is doing all sort of function what is being also available in a regular factory, and that is why the people thought that we can be able to utilize the cell for the cellular factory, or we can use that cell for the producing the different types of products. So, the concept of cell as the production factory comes because the cell is a building block for all the existing creature from the bacteria to human, right. And it is so surprising that a cell can resemble an entire factory itself, its main stations which contains all the information like the nucleus. They encrypt their order in the form of DNA, which can easily be read by the messenger RNA. So, that the DNA and RNA is the cellular code of conduct followed by the RNA into the pain, that is the main factory product. So, in the cell you are actually going to have the main product, that is the proteins, whereas in the factory you are actually going to have the different types of products as per the requirements. The protein is then going to be processed to the endoplasmic reticulum for the quality check, and if everything is process is all right, it is going to be packed into the different types of packaging material, and that is being done into the Golgi body, and then it is going to be ready for supply. Finally, all factories have their own source of energy, and the bacteria or mitochondria or the chloroplast is going to do that function. The cell factory engineering is a process of rationally modifying the cellular factories to produce the desired molecule of sustainable economical and scientific interest. These products can include the native or the foreign proteins, native metabolites or molecules from the completely new biosynthetic pathway. So, cell is capable of producing the proteins, cell is capable of producing the metabolic products, right. So, it is actually going to produce like different types of metabolic products. Either you can take the product, or you actually can modify the product by utilizing or introducing the different types of enzymes and so on. What is the goal? To turn a cell like a microbe into a high performance production unit for the various type of applications. So, cellular factory is a concept where you are actually going to use the cell as a factory, so that cell is going to produce your desirable product. Why it is there is a need to have the engineered cell factory? Because you are going to have the mass production, and it is going to be more sustainable. So, natural sources of variable compounds are often scarce, or production is too low for the commercial uses. The engineering a cell factory allow for the large scale controlled production. So, one of the major issue with the natural product is that their quantities are very low, and they are also present with the other molecules, right. So, the purification and the other problems are always been there, right. And if you want to take out these small quantity of molecules from the intrinsic or from the natural sources, it becomes commercially not viable, because they will be able to their purification and upscaling and all those kind of things will actually increase the cost of the production. So, as an alternative, what you can do is you can actually produce the product into a cellular factory, where you are actually going to give the instruction to the cell, so that it will actually going to produce that particular product in a very large quantity. And the other point is the sustainability. So, we can actually create a renewable petroleum based chemical and non renewable sources, and sustainability is also one of the important component, because if you are going to utilize the cellular factories, they are actually going to be more sustainable, because they are going to use the natural products, or they are actually going to use the natural sources. And for these purpose, you can actually be use these cellular factories for producing the vaccines. You can be able to use that for producing the monoclonal antibodies. You can use that for producing the bioplastics, biofuels, and then also you can use this for different types of tissue engineering applications. And the other third point is the precision. So, we can design the cells to produce the molecule with the specific properties, such as therapeutic protein with the enhanced specificity, and then you also going to have the, instead of harvesting a rare plant for the drug, we can engineer a common microbe to produce the same compound efficiently in a lab. So, in the cellular factory toolbox strategies, what you are going to do is the cellular strategies can be grouped into three main groups based on the type of product being modified. You can have the native metabolites, you can have the heterogeneous pathways, or you can actually have the protein expression. So, depending upon the type of metabolites you are trying to produce into these cellular factories, you can be able to have the different types of toolbox. So, boosting the native metabolites. So, if the goal is to make the cell produce a compound it naturally synthesize. How it is going to work? So, you are going to do the overexpression, increase the number of key enzyme into the biosynthetic pathway of that particular metabolites. Then you require remove the bottlenecks. So, you delete the competitive pathway that use the same starting material. So, for example, if you are trying to produce the pyruvate, right, if you are trying to generate the large quantity of pyruvate, right, and you know that the pyruvate will come from the glucose by a process called glycolysis, right. So, if you are trying to generate the pyruvate, right, first thing what you have to do is you have to increase the concentration of the glycolysis enzyme, so that you are going to produce more and more pyruvate. But once the pyruvate is being produced, it is actually going to get into the Krebs cycle, and that is how the Krebs cycle is actually going to use. It will get into the Krebs cycle, and that is how whatever the pyruvate you will produce, it will enter into the Krebs cycle, and eventually it is going to be used. You have to do something so that it is not going to be the case. It is actually going to remove, it is going to accumulate, right. And then you also going to ensure the degradation prevention. So, you are also going to ensure that the pyruvate is going to be remain stable, it should not be get degraded, right. So, for example, engineering the function asparagine Niger's to increase the metabolic flux through its glycolytic pathway to boost the citric acid productions. Then you also have the new assembly lines. So, you can actually use the heterogeneous pathway. So, goal is to enable a cell to produce a compound it has never made before. So, this is going to be the exogenous molecule what it is going to produce, right. So, in the previous examples we have seen where the cell is capable of making a particular product. So, we have enhanced the protein that particular production of that particular product simply by increasing the pathways enzyme, and also we have done the blocking so that it produced biometabolites should not be utilized into the other pathway, and we also reduce the degradation also. In the other one, in this particular strategy where you are actually going to have the heterogeneous pathway, you are going to introduce a particular gene. So, what you are going to do is you are going to introduce a gene cluster. So, identify and transfer the entire gene cluster to that code all the necessary enzyme for the new pathway, right. So, in this case the cell is intrinsically is not producing that particular molecule. So, what you are doing is you are putting the whole pathway into that particular cell, and then you are doing the host optimization. So, ensuring that the new pathway is compatible with the host cell's internal environment, like the providing necessary cofactors for or the substrates. Example is you are going to introduce a gene for the anti-cancer drug Taxol from a rare plant into a endophytic phosphate to allow for the large scale productions. Or number 2 is engineering the yeast Pichia pectoris for a gene from a fungus to produce the gamma linolenic acid, a fatty acid used into the nutrition. So, these are the 2 examples where you are actually putting the complete pathway into the cell, and then you are asking the cell to produce that particular biometabolites. Then you also have the strategy number 3 where you are going to use the expressing the specific proteins. So, goal is to produce a single specific protein for a particular use. How it is going to work? So, you are going to do the promoter engineering, right. So, using a strong on switch promoter to drive the high level of gene expressions. You can also do the secretion. So, engineering the cells to efficiently secrete the protein, making it easier to purify, or you can do the post translational modifications. So, modifying the protein after it is made, for example, by adding the sugar molecule or the glycosylations or phosphorylations or acetylations. You can do all these kind of things so that you are going to produce a functional protein. Example is producing the therapeutic protein like the insulin or the monoclonal antibody using the engineered yeast or the fungal cell. So, in an insulin production, how the things are going to work? So, you have the bacteria, you are going to use the bacteria. So, what you are going to do is you are going to make a recombinant plasmid, right, where you are actually going to put the insulin gene, and you are actually going to produce the insulin, so you are taking out the insulin gene from the beta cell, right, and you are putting that into this particular plasmid using this particular multiple cloning site, and that is why you have produced the recombinant clone. And then you put that into the bacteria, and bacteria is going to express the large quantity of the insulin, and then you can actually be able to do the purification and extraction using the different types of chromatography technique, and that is how you are going to have the insulin. What are the key players into the cellular factory or cellular factory engineering? So, the first is the host, right. So, you can actually have the different types of host. You can have the bacterial host, you can have the yeast stores. So, often used for the fast growth and simple genetics. E. coli is a common workhouse. Then you can use the yeast. So, fungi are the single cell like the Saccharomyces cerevisiae are great for expressing the protein as the well understood genetics. Then you can use the filamentous fungus. So, you can use like these are multicelled fungus like Aspergillus and Trichoderma, and they are the hyper producer and excellent secretion of enzyme, making them ideal for the industrial applications. And then you can also use the mammalian cells. They are used for the insulin productions, hormones, erythropoietin, and so on, right. And then they are used for growth factors and the blood clotting factors. So, depending upon the type of product you want to use, the first thing is you have to choose a particular host where you are actually going to do the production part. Then the second thing is the applications, right. So, it is these cellular factories are actually going to have the extensive applications in the different types of areas. So, in the industrial applications, you can have to you can be able to use this for producing the pharmaceuticals, you can use this for producing the different types of enzymes, you can use this for producing the biofuels and biochemicals, and then you can also use for producing the natural products. So, you can use this for producing the therapeutic proteins like the insulin or the monoclonal antibodies, or you can use this for producing the different types of enzymes. For example, you can use this for producing the enzyme like lipase, horseradish peroxidase, catalase, and all those kind of things, and they are being used in different types of industries like the food processing industries and the biofuel production. Then you can also use this for biofuel and biochemicals. So, engineer microbes to produce the ethanol, biodiesel, and bulk chemical like the adipic acid. And then you can also use this for biosynthesis of the antibiotics as well as the vitamins. Then we also have the application in the research field. So, you can actually use the functional genomics, you can do the drug discovery, you can use this for medical and therapeutic applications like where you can actually use this for producing the prebiotics or the therapeutic proteins. So, in the research applications, you can study how the genes and the protein works by engineering them in a controlled environment. For the drug discovery, you can use the engineered cell to screen for the potential new drug candidates. Like for example, you can use that for producing the random molecules, and then these random molecules can be extracted from the particular host, and then they can be tested for the different types of applications. Then it can also be used in the medical and therapeutic applications where you can use the engineering prebiotics to produce the beneficial product directly inside the body. So, basically what you can do is instead of giving the prebiotics which is already been purified, what you can do is you can give them a bacteria or I will say recombinant bacteria which actually is going to produce the different types of prebiotics. Then you can also produce the protein with the enhanced quality for the pharmaceutical applications. So, one of the example is recombinant protein production with the filamentous bacteria. And what is the challenge? The filamentous bacteria are excellent at secreting protein, but they are also produce protease that degrade the desirable protein. Additionally, their glycosylation patterns are different from the male, which is a problem for the human therapeutic proteins, right, because the glycosylation pattern is actually going to work as because they are actually going to generate the antigenic response, and because of that these proteins probably will cause the inflammation or the other kinds of immune reactions. What is the solution? The solution is you can do the protease deficient host. So, scientists have created a mutant strain of fungi like Aspergillus niger that are missing the gene for degrading the protease. So, it is actually minus protease strain what you can actually generate, and because of that it is not going to degrade the proteins. Then you can also have the fusion proteins, they are also called gene fusion strategy, where the target protein is fused to a highly secreted fungal protein and which improves the secretion and the stability. And then you can also have the humanized glycosylation. So, research is going on to engineer human like glycosylation pathway in fungi to make them more suitable for producing the human therapeutic proteins. Then we also have the trichoderma and biofuel product connections. So, trichoderma is an exceptional producer of cellulases and the hemicellulases, which are key enzyme for the breakdown of the plant biomass. And the industrial uptake, it can have the hyper producer over 100 grams per liter of the extracellular protein, which matches of the cellulose. This make it a principal target for creating the cellulose derived ethanol as a biofuel. And then you can use this for the biocontrol agents. So some trichoderma species are also used in agriculture to produce the plant from the pathogen by attacking other fungi. What are the challenges and the future directions? So, what is the challenge? You can actually have the low transformance rate. So, it can be difficult to get the new genes into some of the fungi. Then you also have the protein degradation. So, as mentioned earlier, the host protease can still degrade the target proteins, and you can also have the glycosylation. So, mismatch in glycosylation pattern for the human therapeutic protein remains a challenge, and this is already people are working where they are introducing the new glycolytic pathway enzyme, so that the glycosylation pattern what is being found in the fungi is could be as good as the glycosylation pattern into the mammalian system. And what is the future direction? So, you can actually be able to use the better genome editing tools, like for example, you can use the CRISPR-Cas or synthetic biology. So, use gene editing tool like CRISPRs are been making it easier to precisely modify the genome, and you can do the system level design. So, instead of modifying one gene, scientists are now using the computer model to design and optimize the entire metabolic systems. And then you can also do the microbial consortia. The future may involve the engineering multiple different microbes to work together in a complex system for a specific task. And then you can also use the engineered fungal pathogens. So, you can actually use like, so fungi cause the significant disease in humans, animals and plant, leading to a major economic and social cost. And what is the opportunity? Opportunity is that you can use recombinant technology to understand the molecular mechanism behind the fungal pathogenesis. Like for example, the scientists are using the engineered rhizopus to understand how its secreted protease contribute to its virulence as a pathogen, and this knowledge can lead to a new drug that target this specific virulence factor without harming the host. And then you can also study the biosynthetic pathway in the other host, right. So, the fungi are the enormous source of the novel biosynthetic pathway with an estimated of 1.5 million species, most of which have not yet studied. Many natural fungi hosts for these pathways are not suitable for the large scale manufacturing, and the solution is the transferring these valuable biosynthetic capabilities to other more manageable hosts like fungi, bacteria or even plants. There is an example like transferring the gene for the highly unsaturated fatty acid from the fungi into a canola plant to create new oilseed crops. Then you can also do the engineering for the environmental solutions. So, for example, the lignin is a very important metabolite what is present into the plant, and that makes the grass little hard to digest, right. So, this is a bottleneck for the biofuel production. So, what you can do is you can actually generate a fungal strain. So, you can actually generate like white rot fungi, like the this fungi are only the microbe capable of efficiently degrading the lignin without attacking the cellulose. And what you can do is scientists are studying the unique enzyme from these fungi to transfer these capabilities to more conventional industrial host. Examples are lactose enzyme from the white rot fungi have been expressed as a recombinant enzyme in Aspergillus oryzae and are active against the pollutants. Cell factory engineering as a paradigm shift, right. So, from single product to a complex system, the field is moving beyond just making a single protein to engineering entire metabolic pathway and multi-protein complexes. From one host to many, we are not just using the one host organism, we are transferring valuable capabilities from one species to another to find the best production platform. From manufacturing to beyond, right. So, applications are expanding beyond manufacturing to include the understanding and solving the complex biological problems like the pathogenesis. So, this is all about the cell as the the production factory. We have discussed about the different strategies, how you can be able to use that for producing the different types of products. So, we can actually take the product which is been a part of the metabolic pathway. So, we can actually be able to enhance the production by increasing the pathway enzymes, or we can actually be able to reduce the degradation. The alternate strategy could be that we introduce a complete pathway into a particular organisms and produce the product of our need. And the third strategy would be that you are actually going to exogenously express the particular protein, so that it is actually going to give you the large quantity of that particular product. So, these are the some of the the strategies what you can use, where you can be able to exploit the cell as a production factory. And the advantage is that since it is a biological in nature, it is a natural, you probably will have the better product without even having the much effort to be put in terms of purifying the particular product from different types of the indigenous product and so on. And then at the end, we also have the flexibility of modifying the product as per your need actually. So, with this I would like to conclude my lecture here. In a subsequent lecture, we will discuss some more aspects related to cell and molecular biology. Thank you.